Hey folks, and welcome to another edition of Zog Science. We're going to be starting our little section on macromolecules, and we're going to be getting into our first sets of macromolecules, which are lipids and carbohydrates. So let's talk about what macromolecules are to start off with. These are big, complex molecules that accomplish life's functions, and they are broken down into four main groups: carbohydrates, lipids, proteins, and nucleic acids. Now, carbohydrates, proteins, and nucleic acids are a little bit different from lipids in that they are all polymers, poly meaning many, and they are composed of long chains of individual monomers. So when we have polymers being created, they are formed through what is called a dehydration synthesis reaction, where a water molecule is removed between two different monomers, and it is basically kicked out, and then a bond is formed between those monomers. We can also have a hydrolysis reaction, which is what is used to break down polymers, where we add water back in, and then now that breaks it apart. This builds on what we've been talking about in terms of water being very important, right? Because water is used to both formed during the synthesis of polymers and then is also used in the breakdown of polymers. And since polymers make up the majority of macromolecules, it's very important that we have these. Both of these reactions are mediated by enzymes, and enzymes are special types of proteins, which we'll get into more in our next section. So some things to focus on as we're kind of going through these four different macromolecules. I want to focus on the structure, the function, the elements that are a part of each of them, the monomer, and then the polymers that are a part of it. So let's talk about carbohydrates first. Carbohydrates many times are known as sugars or starches. These are made up of carbon, hydrogen, and oxygen exclusively. There are no other elements found. And generally, they're going to be in a 1 to 2 to 1 ratio or something very close to it. There are carbohydrates that don't follow this 1 to 2 to 1 ratio specifically. Monomers are a lot of times called monosaccharides or single sugars. And then the different sugars can have different numbers of carbons. So basically, when we talk about carbohydrates, we're talking about chains or rings that are linked together. And you can see that we have lots of different types. We have trioses, pentoses, hexoses, and again, these can all be have different locations of where the functional groups are. So, for example, glyceraldehydes are different from dihydroxyacetones. It all has to do with where this double bonded oxygen is, right? This double bonded oxygen is on the end, or if it's in the middle, and that's going to affect the function of that carbohydrate. And so you can have the same number of carbons, but having that double bonded oxygen in different places is going to affect the structure and therefore the function of it. Monosaccharides and disaccharides are single sugars. These are your monomers or dimers, as these are called because there are two of them. And they tend to be used for quick energy, and they're usually found in a ring form. They're certainly originally created in a linear form, but they are put into a ring form pretty quick after that, and that's where they spend the majority of their time. A lot of the really well-known hexose sugars are glucose, galactose, and fructose, and we can combine those in different orders in order to form our different dimers, such as maltose, sucrose, and lactose. I'm sure most of you have heard of lactose. Sucrose is your simple table sugar, and maltose is found in a lot of plants. And so it's all about how these are different monomers are kind of linked up to form these disaccharides, and again, that is formed using that dehydration synthesis reaction. Polysaccharides are different monosaccharides or disaccharides strung together, and their two main functions are either for kind of, again, short-term energy storage or for structural support. Animals use mainly glycogen, and that glycogen is used as a long-term energy storage, while plants mainly use starch, or also known as amylose, for energy storage. And then plants also use cellulose to help build their cell walls and provide structure. Now, it's interesting because when we take a look at starch versus cellulose, starch and cellulose actually are both made up of just long chains of glucose molecules. However, starch is what we call an alpha linkage, and an alpha linkage has all of the alcohol groups on the same side of the chain, whereas the beta glucose linkages are where the alcohol groups is actually flipped. And this is very important because as we will see, cellulose's beta chains cannot be easily digested by enzymes found in animals. So we as animals don't actually have enzymes that allow us to break down those beta linkages. We can break down the starch just fine, but cellulose we can't really do. And so what that means is that we have evolved different ways to actually be able to deal with this. Termites have symbiotic relationships within their guts with some protists, and these protists do actually have the enzymes that they need in order to be able to break down those beta linkages. Cows and other ruminants have an expanded GI tract, and this is when we talk about cows having 4 stomachs, and as well as the ability to regurgitate their food. And so what this does is it gives them more time to be able to break down the cellulose. While we don't have enzymes that are specifically built to break down the cellulose, they can slowly do it given enough periods of time. So what these cows do is they bring the grass into their mouths, they kind of swallow it down into their stomachs. It goes through the stomachs, picks up some enzymes, and then they kind of regurgitate it up into their mouths, spend some more time mechanically digesting it, chewing on it, and then they send it back down, and they just kind of go through this process. And so by doing so, they're able to eventually extract enough nutrients to be able to live based off of that. We also have some other animals that have some different techniques for doing this. We've got rabbits, and the rabbits, what they do is they actually will, eat the grass, goes through their GI tract, they excrete it as feces, and then they will eat the feces and put it through their GI tract again. So again, the idea is that they don't have enzymes that are specifically built to break down those beta chains. However, by sending it through their GI system multiple times, they're able to extract enough nutrients. Now, we as humans, we don't really have a lot of those kinds of interactions. What we use the cellulose mostly for is to help with digestion. So when you look on a package of food and you see that it's got dietary fiber, that's really just telling you that it's got cellulose. And that cellulose, what it does is it helps to kind of rub against the inside of your GI tract. And when it rubs up against the inside of your GI tract, your body actually releases mucus along there, and that helps to lubricate the GI system and helps keep you regular. And that's why people, with low fiber tends to get constipation because they don't have enough lubrication inside their GI system, helping to kind of keep things moving along. All right, so that was all we talked about all you need to know about carbohydrates. Let's talk about lipids, all right? Lipids are not technically polymers, so they don't really go through this whole dehydration synthesis reaction thing where they kind of add a whole bunch of things on. And what makes them kind of unique is that they are hydrophobic or non-polar. They're made up of mostly carbons and hydrogens with only a little bit of oxygen, which makes sense when you think about them being non-polar, right? Because the oxygens are what make a lot of other molecules polar, or we'll also see that nitrogens and phosphoruses also do the same thing. But being mostly made up of carbon and hydrogen, we get a lot of non-polarity found within lipids. There are 3 different kind of main types of lipids, and the first ones are the fatty acids. Fatty acids can be known as triglycerides, although they're not always in the triglyceride form. Sometimes they are found in different forms. And they're made up of a glycerol and a fatty acid. The glycerol is just kind of like a carbon chain with a couple of oxygens, and then a long chain of fatty acid, which is just a carbon and hydrogen chain. These are mostly for long-term energy storage and insulation. It's good that we have some fat in our bodies because it helps to keep us warm. And these can be either saturated or unsaturated. Saturated fats only have single bonds, and they're completely packed with hydrogen. And what that means is that they can actually pack in very tightly with one another, meaning that they become solid at room temperature. They're not spread out. They're pretty close. And these are all found in animals, which is good. You want to be able to move around a little bit. And we don't really have unsaturated fats in our bodies very much. Saturated fats also tend to have a higher energy content because you've got more hydrogen bonds, and those hydrogen bonds have more energy than a double bond does. Unsaturated fats, these have some double bonds, and what that means is that it actually puts little kinks in the fatty acid chain, and because it's got a kink in it, it doesn't allow the molecules to become as tightly packed with one another. They spread out a little more, and so therefore these are all liquids at room temperature. An example of this would be olive oil. So olive oil is an unsaturated fat. It is still a fat, but it's unsaturated, and because of those kinks in the chain, it actually makes it keeps it from becoming a solid like something like butter, which is found in animals or is a product of animals. One of the other main types of lipids are phospholipids, and phospholipids are a major component of cell membranes, and they are composed of a hydrophilic head and two hydrophobic tails. And what happens is that when you put them together, they automatically form a lipid bilayer. So the hydrophobic head, the hydrophilic head consists of a phosphate group, and there you've got a whole bunch of oxygens and the phosphorus, and that becomes very polar, and therefore it likes water. It likes to hang out with the water. And then you've got a hydrophobic tail, which again is a long chain of carbons and hydrogens. And so those are hydrophobic. They don't really like to be around water. So what happens is that when you place all these together, they're automatically going to, if you have water there as well, they're automatically going to line up where the hydrophilic head is towards the water, hydrophobic tails are lined up with each other, and then the other hydrophobic tail is on the other side. Well, this is very good because now you've created a barrier between an outside and an inside, and this is what we believe is the basis for how the first cell membranes formed, right? We had these phospholipids, they kind of congregated somewhere, and eventually they kind of sealed off and made themselves into a little sphere from which they could then, evolve other things from there. We'll talk more about that when we actually get to our evolution. But this makes for a kind of interesting point, which is that water has a very hard time getting across a cell membrane, right? Because in the middle of the cell membrane is this hydrophobic region, and water cannot cross through that hydrophobic region. And we'll talk about how cells deal with that when we get to our cell unit next. Steroids are the final major class of lipids. These are identified by having a carbon skeleton composed of 4 rings. Many times it's going to be 4 six-sided rings and 1 5-sided ring, although there are other configurations that those can take as well. And the function is determined by what functional groups are actually attached to the rings. So we can have lots of different ones attached in different places, and they all, it just completely depends on where they're located, right? Again, form and function. Most steroids are chemical messengers, and that's because they are nonpolar, right? There's not a lot of polarity here, and therefore they can easily dissolve across that lipid bilayer. So they're able to move across the lipid bilayer and get into cells easily, and therefore they can carry out their function relatively easily. This is why steroids are very high impactful drugs. Whether or not you're taking like a steroid for some sort of inflammation, or whether you're taking like a steroid to actually buff up, like things like that. Steroids are high impact because they can very easily get into cells, and they can easily get into the DNA and help it change expression, or whatever it is that they're doing. They're high impact. They're able to carry out a lot of functions. So anyways, that's all we've got for you today on lipids and carbohydrates. Next up, we'll talk about proteins and nucleic acids. Thanks for watching, and I'll see you next time.